further, let us discuss the structure of pollen grain, or it is nothing but microspore. Now microspore, these are the produced from microspore mother cell by meiosis. So microspores are obviously haploid. Each microspore is unicellular and uninucleate. So microspore is haploid, unicellular and uninucleate structure. They are either spherical in shape or oval. So shape may vary from plant to plant. They are either spherical or oval. Now let us discuss the structure in detail. Suppose this is the single cell of microspore or pollen grain. It is surrounded by two layers. The outer layer is thick, cutinized and hard. It is called as exine. The inner layer is thin, flexible and elastic. It is called as intine. Exine and intine, these two layers are collectively called as sporoderm. Spore here indicating the microspore. Derma means the skin, the protective layer. So here sporoderm consists of two layers. This question can be asked. So outermost is called as exine, inner is called as intine. Exine is thick, hard and cutinized, and intine is thin, flexible and elastic layer. Inside this there is some amount of cytoplasm, very scanty, and a single nucleus. As we have discussed, it is uninucleate structure and this nucleus is haploid. So this is the very simple structure of pollen grain. Now important thing is the exine shows characteristic thickening. As you have studied, it shows presence of a toughest biochemical material which is called as sporopollenin, and this sporopollenin is non-degradable by any enzyme or any different chemicals. So therefore pollens can be stored or they can be traveled to a long distance without any damage. But this exine is absent or thin at certain places. Let us discuss. This is the region of thin exine, or here exine is absent. This region of thin exine or exine which is absent in this particular region is called as germ pores. So in this pollen grain, how many germ pores you observe? 1, 2 and 3. When three germ pores are present, then this is taxonomically important because you can identify while you observe a microspore or pollen grain under microscope. When you observe 3 germ pore, then it will be of dicot plant, and that is for sure. So number of germ pore is helpful taxonomically. Or when a single germ pore is present, at that time it belongs to monocot. This part is very important. Again, when 3 germ pores are present, the microspore is called as triporate. Means 3 pores are present. We can ask in the question, triporate pollen grain is the characteristic feature of dicot, obviously, and here uniporate means having single germ pore is the characteristic feature of monocot. Now see, suppose this is the pollen grain, then you can observe the germ pore like this. A small pore is present, or 1 or 3 pores are present there. Sometimes the pollen grain show the germ pore in the form of a furrow like this. Now this is called as pore and this is called as colpa. So when pores are present, it is called as triporate, and when colpa is present, at that time it is called as tricolpate. So tricolpate or triporate, both are characteristically seen in dicots. When single colpa is present, at that time it is called as unicolpate or monocolpate. When single furrow is present, single colpa is present, then you can call it as unicolpate or monocolpate, or you can also call it as uniporate when pore is present, but both are characteristically seen in monocots. Now this is important in the taxonomy. Again, the composition is very important. As we have discussed, the Exine is made up of sporopollen. It is not entire. It is thin or absent at certain places, which is called a germ pore. Now let us discuss the structure or composition of intine. It is thin, elastic and flexible, and it shows the presence of, or it is made up of, two components, and that is cellulose plus pectin, which can collectively be called as pectocellulose. In your MCQ question bank or MCQ questions, the option is like this: pectocellulose. Then you can get confused. Cellulose plus pectin can collectively be called as pectocellulose. So intine, it is cutinized and made up of sporopollen. Intine, it is thin, elastic, and it is made up of cellulose and pectin. So this is the entire structure of a pollen grain or microspore. Now we are going to discuss the development of male gametophyte. This process is also called as microgametogenesis. The process of formation of microspore is called as microsporogenesis, and the process of development of male gametophyte is also called as microgametogenesis. Now microgametogenesis is a process of formation of male gametophyte from a single microspore. Now it is very clear that the process of microgametogenesis occur from a single cell, which is called as microspore. And therefore, microspore is the first cell of male gametophyte. The entire development of the male gametophyte occur inside the microspore. It will not seen outside of the microspore, and therefore, or you can say, entire process of microgametogenesis or development of male gametophyte occur within the boundary of sporoderm, occur inside the microspore itself, and therefore development of male gametophyte is termed as endosporic, which is occurring inside the microspore. And development of male gametophyte from single microspore requires two mitotic divisions. These two concepts are very important, that microgametogenesis or development of male gametophyte is endosporic, and it requires how many mitotic division? That part is very important. It requires two mitotic division, two successive mitotic division for a development of male gametophyte from a single microspore, which is the first cell of male gametophyte. Let us discuss how it occurs. Development of male gametophyte is achieved in two different steps. The first is called as before pollination, and the second one is called as after pollination. Before pollination, the microspore or pollen grain, they are present inside the anther, and therefore you can say before pollination inside anther. After pollination, the microspores are released outward, and those will reach up to the female gametophyte. It means simply they are approaching the carpel and the part of carpel which is called as stigma, and therefore the second step is called as after pollination on the stigma. Or one more concept you can explain here: before pollination inside the anther. Anther is the native place of microspore, and therefore this development is also called as in situ development, in the original place, in the natural place. And when pollen released out from the native place, the original place, they will come in contact with stigma. Now this development is called as ex situ development. So these two parts are given in your notes. This is very important. Before pollination inside the anther, and after pollination on the stigma. You can call it as in situ, or the second one is called as ex situ. Now let us discuss one by one. This is the single cell microspore having double layered sporoderm, exine and intine. This one is exine. Inner is called as intine. the single nucleus is present inside this, and this nucleus is haploid. Now, the microspore shows first mitotic division. So you can say first mitosis, and this first mitosis is unequal in nature. Means this single cell divide unequally to produce two unequal cells like this. This is the sporoidal boundary. Entire process is occurring inside the microspore, and therefore we use the term endosporic development. Now here two cells are formed. This is the first cell, which is large, and this is the second cell, which is small. This cell is called as tube cell or vegetative cell, which is large one, having a distinct wall and a single haploid nucleus after first mitosis. This is the second cell. It is called as generative cell or germ cell. Now this cell is small in size, and therefore I have used the term unequal. So microspore divide by first mitosis to produce two unequal cells. One is large, it is tube cell or vegetative cell, and the second is called a generative cell or germ cell. Now this generative cell or germ cell, it lacks distinct wall, then reserve food and vacuoles, they are absent in the generative cell. Now here there is a distinct wall to the tube cell or vegetative cell, but generative cell is freely suspended in the cytoplasm of tube cell. It lacks distinct wall, reserve food material and vacuole. They are completely absent. So this is the first stage, the first mitosis. As we have discussed, development of male gametophyte requires two mitotic division. Out of that, one is completed, and therefore this structure is called as partially developed male gametophyte. Partially developed male gametophyte is having two cells. One is called as tube cell, another is called a generative cell, or you can use another terms like vegetative cell and germ cell. In most of the angiosperm, say for example, about 60 to 80% of angiosperm, the pollen grains are released at this particular stage, which is called as two-celled stage. And in remaining angiosperm, that is 20 to 40% of angiosperm, they follow the second mitotic division inside the anther itself, or in situ development. They produce three cells and then they release their pollen grains, and that is called as three-cell stage. But which is the most probable mechanism, most common mechanism? Many angiosperm, they used to release their pollen grains at a two-cell stage, and that is the common one. Now the next development, the second mitosis, will occur after pollination on the stigma, that is by means of XII development. We are going to discuss it. The second step is after pollination on the stigma, when the angiospermic pollen, which is released from the anther at two-celled stage, as we have discussed, it shows presence of a tube cell and a small generative cell. Now this two-celled stage pollen that will arrive to the carpel, and the communicating structure of the carpel, which is called as stigma, and this pollen resides on, settle down on stigma. A stigma secretes the sugary substance which is called as exudate. You can compare like you are entering in the house of guest is arriving at you, then you will offer tea. Here the pollen is like a guest and stigma is offering the sugary substance. Jokes apart, but that is easy to understand, that sugary substance is secreted by the stigma, which is available for the pollen grain. Now this pollen will absorb the sugary substance or the exudate secreted by the stigma, and internal cytoplasmic content will enhance. Now pollen grain is having the structure of having a wall like this, the outer wall which is called as exine, inner wall which is called as intine. And this exine, at certain places it is absent, or it is thin like this, what we call it a germ pore. When internal cytoplasmic content will increase, that will exert a pressure on the outward direction. Exine is non-responsive to the pressure because it is tough, hard, cutinized, and the pressure is entirely exerted on the thin and elastic wall layer, which is called as the intine. And so, due to excess pressure exerted on intine, the intine will come out in the form of a tube, and this structure is called as pollen tube. Now you can understand why this cell is called as tube cell, because the boundary of the cell of this tube cell is responsible for formation of pollen tube. So when pollen absorbs the sugary substance or exudate, it will form the pollen tube, and this pollen tube starts growing from stigma towards the anther. From stigma towards the ovary. It is like this. So formation of pollen tube is after absorbing the sugary substance. Now one question can be asked, the pollen tube is made up of, or what is the composition of pollen tube? Now composition of pollen tube is exactly similar to that of intine, and intine is made up of cellulose plus pectin, and therefore pollen tube is also made up of cellulose plus pectin. As simple as that. Now see, the structure is like this now. This is the exine. This one is intine. Due to pressure exerted from inner side towards outer side, it is coming out in the form of pollen tube. Now this is the tube cell, and it is having the generative cell. Now entire content which is present inside the cell, inside the microspore, that will come out into the pollen tube in a particular sequence. That sequence is very important. The first structure which will come out is called as tube nucleus, then cytoplasm of the tube cell, and it is followed by a structure which is called as generative cell. So when pollen tube is formed, the first structure which is entering inside the pollen tube is tube nucleus, which is followed by the cytoplasm. Basically, we have discussed that cytoplasm which is present inside the microspore is very small, is very scanty, and therefore cytoplasm which is present in the tube cell is a vacuolated cytoplasm having gaps in between, which is followed by the generative cell which is entering last in the pollen tube. The first mitosis has already occurred, now the second mitosis is remaining. Now this generative cell is following the second mitosis. This generative cell is lacking distinct wall, no reserve food material is there, no vacuoles are there. After second mitosis, this generative cell divide to produce two cells, two haploid cells, and now the structure will appear like this. Again, this is exine, this one is intine, pollen tube is coming out like this. At the tip of pollen tube there is tube nucleus, which is followed by the vacuolated cytoplasm, and this generative cell divide by mitosis to produce two haploid male gametes. These are the haploid structure. Now the entire development of male gametophyte is completed. The fully developed male gametophyte is a 3-cell structure, and partially developed male gametophyte is 2-cell structure. Whenever the diagram is asked in your exam, write a neat label diagram of male gametophyte, then you have to go for the last diagram which is given here. And when they ask, describe, or you can you have to draw the diagram of development of male gametophyte, then you have to start with pollen grain, then the 2-cell stage, then last the fully developed male gametophyte. So this process is called as endosporic. Now you can ask the question, because as we have discussed, the process is called as endosporic. Entire development will occur inside the microspore, inside the cell. But some of you can ask the question like this, because the pollen tube is coming out of the cell, but remember it is itself in the boundary of intine, and intine is the component of wall of microspore. So none of the cell is going outside of the microspore, and therefore development of male gametophyte is ideally called as endosporic.